two things. First of all, the arrival of Anna Yopart in my laboratory, a molecularly trained postdoc who did stuff I could never do. And second of all, this review of our first paper on these species from a reviewer. We said, this is for evolution, by the way. That paper was initially rejected because of this review that said, well, we're not working on two species. What we're working on is two populations. I mean, this, despite the fact that they differ morphologically, they have 12 forms of reproductive isolation between them, that they maintain their morphological distinctness and sympathy. Some crazy reviewer said, well, they're not different species, so you're not working on speciation. So we of course, argued cogently to the contrary, pointing out the salient facts, and the paper was accepted, but this led us to wonder, well, yeah, how much integration really is there between the species, because they live together in the same country. And so we did a molecular analysis, and when I say we, I mean Anna. That's what happens when you get old. We did a transect from sea level up to the top of the mountain to sea, and looking at flies at each stage of the elevational gradient to see how much gene flow there actually was between these two species. Okay. How do you do that? It's kind of tedious population genetic analysis, of which I'm only barely competent to understand, much less do. The way you measure integration between two species that live in the same area depends on the region of the genome you're looking at. If you're looking at regions where there's normal amounts of recombination, then you look at the level of shared polymorphism between those two species. If species A has two nucleotides A and B, and there's integration, then species B should also show both of those polymorphic forms. So you look at the shared level of polymorphism. Of course, you can get that not just from integration, but from inheritance from a polymorphic ancestor. So you have to do various coalescence simulations and statistical analysis to rule out ancestral polymorphisms and single in only on those regions where the polymorphisms that are shared really represent integration from one species to another. For regions that are non-recombining, where you cannot, there's low polymorphism, so you can't do this kind of test. You simply have to look at the level of genetic divergence between the two species in those regions, and compare it to, and this is where we're fortunate, you compare it to another pair of species where there's no possibility of integration because they live in different places. Okay, so we measure genetic divergence in these regions between Yucuba and Santa Maria, and we compare them to what we call our control species, which we're so lucky to have these, Drosophila seychellia and Drosophila mauritiana. These species are also 400,000 years old, but they're endemic to different islands in the Indian Ocean. So they've never encountered each other. So we can use this as control, look at the genetic divergence between these two, and compare that to the genetic divergence between the two species of the same age that actually have the opportunity to exchange genes. So we can single out those regions where there has actually been integration. Okay. And so we looked at 29 regions. Actually, I talked to Anna yesterday. I think it's actually more than this. Scattered throughout the genome, looking for integration. Some regions of low recombination, for example, the mitochondria, Y chromosome tip of the X, and other regions of high recombination. There's 29 of those, so we looked at that. And I'll just tell you the results in general. For regions of low recombination, we looked at genetic divergence. Here's the pair of species we're looking at to see if there's integration. Yucuba and Santa Maria, looking at two regions of low recombination, the Y chromosome and the nucleus. And here's the control pair, the same age. And you can see right off the bat that either both for the Y chromosome and for the nucleus, the genetic divergence between these two pairs of species is pretty much the same. These are completely allopatric. They've never had a chance to exchange genes. These have a hybrid zone. Nevertheless, the genetic divergence is the same. What that means is that there has been no effective gene exchange in these 9 regions of the genome. So there's not much integration going on here, despite the fact that they do live in the same area. But if you look at the mitochondrial DNA, God, I hate mitochondrial DNA. I really do. And yet everybody works on it. Look at the difference here. There's 65 times more divergence in the control allopathic pair than there is in the pair on the island. In fact, the mitochondria of the two species on the island are identical, in effect. There are no fixed differences between them. They're more identical to each other than mitochondria of Yucuba from the island and Yucuba from the mainland. The mitochondria are in effect identical. What's happened here? Well, you probably know. This is a case of mitochondrial integration. The mitochondria have moved from Drosophila Yucuba into Drosophila Santa Maria. Okay. That gives us a phylogeny, if you look at mitochondria alone, that makes it look like speciation with symplectic, right? Because they're identical. So if you only looked at that top line, you'd say this is a case of symplectic speciation. Look at all the data, you're you realize that it's a case of double colonization. So we have this big disparity here. And if you look at the total genome as a whole, we see extensive introgression of mitochondrial DNA, and we see a little bit of introgression, not so much, in one other non-recombining region, which is the tip of the axon, the cellular gene here. But of the 29 genes that we've looked at, there's really only extensive recombination or gene flow in one region, which is the mitochondria. It's been completely replaced in one species by the other. These other have a statistically significant introgression, but not so much. So what we see here is we don't see islands of speciation. We see islands of introgression. There's small regions where there's been a little bit of leakage of genes between the two species, but by and large, those two species have maintained their genome, and the face of hybridization is largely separate. Okay. So, the conclusions from this: first of all, the mitochondrial DNA of Yakuba has replaced that of Santomea. We know that because of the outgroup on Africa. This is a frequent observation in nature. It's not only seen in Drosophila, or this species of Drosophila is seen in other species. It's seen in mammals, it's seen in birds, it's seen in crickets. There's a paper today showing mitochondrial introgression. Even in plants, with the chloroplast and the mitochondria, there are organelles that introgress pervasively. I think this is another rule of evolutionary biology that has no explanation that I know of. Why do mitochondria move so easily between species, but the rest of the genome doesn't? I mean, people will come up and give me theories, but none of them. And they point me to the old Slatkin paper, which I don't think answers the question. I think that some ambitious young person should first of all compile all the data, and second of all come up with an explanation for why we see this phenomenon over and over again. Regardless of that phenomenon, it's a phenomenon that messes up phylogeny. Okay, so it's an interesting phenomenon. I'm going to show you cannot use mitochondrial DNA to make phylogenies. There's two small nuclear gene regions, introgression. But in contrast to what the reviewer said, who knocked the paper back, these are good biological under any species concept that we want to use. Lineage concept, bimodal species concept, biological species concept, genealogical species concept. There's 25 of them. Under any of those concepts, these are good biological species, I think. I suppose some systemists might say, well, there's some of the genes are paraphyletic, so they're not species, but under any reasonable definition. The last thing I want to talk about before I get to the sort of conundrum I'm going to end with is the sort of ecological differences between the species. Everybody knows that Drosophila do not have ecology. They disport themselves. I'm sure Taylor Simpson pointed this out years ago. They disport themselves in small glass tubes in the laboratory. That's their biology. But no, we know that's not true. It's just hard to study ecology. And we have a case here where actually there's this altitudinal stratification that led us to at least make some very preliminary experiments. Maybe the species differ in their preference and tolerance for temperature. So we just set up these. This is another experiment, which is a post-tenure experiment. We set up, very well. We set up a temperature gradient here, which is, we had the physics department build us this aluminum block, which has partition chambers. We put one end on a hot plate, the other on a cold plate. The temperature varies along that plate from 18 to 30 degrees, which are temperatures that these flies encounter in nature. You put both species in there in these chambers, you let them sort themselves out for half an hour, and then you push the button and they're trapped in one of the 6 chambers. Whose temperature you know because there's a thermocouple in there, and you can measure the preference of these species for the different temperatures in this region. We were astonished to discover that we actually got results from this. Drosophila have no ecology. So here are the temperature preferences. We did 2 strains. We've since replicated this in others. Drosophila yakuba and Drosophila santomea in black and white. Here's the temperature preference. You can see that santomea, both males and females, prefer temperatures about 3 degrees lower. Small standard error, so this is quite repeatable result. So the temperature preference goes along with the altitudinal distribution. If you look at the hybrids, you see the hybrids are intermediate between the two. In particular, and again, pay attention to this, these are the hybrid males that have an X chromosome from Drosophila santomea. These become of interest in a minute. They're intermediate as well. They don't show anything anomalous. The hybrids are halfway between the parental species in temperature preference. They also show differences in temperature tolerance. Drosophila santomea will just die at 28 degrees. It will not hatch from the pupal case. That's the temperature that you regularly encounter at low altitudes on this island. Not only that, but they don't live very long if you're exposed to the high temperature. So, and the hybrids are intermediate again. So we see this not only temperature preference, but temperature tolerance that goes along very well with the altitudinal distribution of these species. I'm sure there are other ecological differences, but they're at least these flies, and we could, find a form of ecological differentiation that might contribute to reproductive isolation, because it keeps the flies at high altitude from going down lower. They wouldn't make it. Okay. So to wind up, so that's basically, that's all the real science. What I want to wind up with, there's another piece of research that was initially rejected by evolution. Now that I'm president, I can say that. But it was finally accepted due to our strenuous and vociferous complaints. This is the study of a hybrid zone between these two species. Now, I showed you this picture before, the altitudinal distribution of these two species. One species gives way to another where the rainforest begins, and there's this hybrid zone at 1,100 and 1,400 meters. But you notice that this altitudinal graph ends at 1,700 meters. The mountain is actually 2,024 meters. So I didn't give you all the data in this graph, and if I did, it would look like this, which is that if you get above 1,500 meters, you find another hybrid zone. In fact, you find a few Drosophila, but at high altitudes, between, say, 1,800 and 2,000 meters, or you can actually go over the mountain to the other side, you find only hybrids, okay? And this is bizarre. This is the only case I know of in the world, maybe some of you know one, in which you find a hybrid zone where the parental species are not there to make the hybrids, okay? So, so how many hybrids are there? There's about 90 actually altogether now that we found. So it's not a lot of them, but we've repeated this observation over 5 years and 2 seasons, wet and dry, and we always find at high altitudes only hybrids. Okay. So this is a mystery in several respects. First of all, the hybrid zone is outside the range of the parental species. It's within the range of Drosophila a little bit, but well above the range of Drosophila, and yet these individuals are hybrids between the two species. Okay, I mean the old saying is it takes 2 to tango, but there's one partner missing. Here. Okay, where do the hybrids come from? Yeah, they have to be made over Drosophila and Santamea, but the Yucuba aren't there. Second of all the hybrids are males. Every single one that we found, up to 90 now, has been a male. Why is that? It's not that we can't see the females, because everything above 1,500 meters that we find, we genotype to see where it is, and we've never found a hybrid female. Despite the fact that we cross these species in the laboratory, you get 50% males, 50% females. In nature, that's in the hybrids. We've never found a hybrid female. Because the question is, where are they? What's going on? Why are they missing? They're extremely viable in the lab, but not so much in nature. Okay. That's another mystery. By the way, I have no answers to any of these questions. You will have answers to these questions, and you will be wrong, because I've given this talk lots of times. We tested almost every possibility. Including genotaxis, stereotaxis, all this kind of stuff. All but 3 of the hybrid males are F1 males, and those 3 are backcross males. So they're all just F1 hybrid females. Where's the rest of the backcross males? Well, maybe it's because the F1 females are missing. Those backcross males, by the way, were found at high altitude, and they're backcrosses to Drosophila yucuba, which is the fly that's not present at high altitude. So it's a similar mystery. And to me, this is the biggest mystery of all. Every single one of those F1 males comes from the cross that's almost impossible to make in the laboratory. It's the cross from Drosophila Santamea females to Drosophila yucuba males. The other cross goes like gangbusters. This one you have to work hard to get it, and yet every single F1 male comes from that impossible cross. So of course the question is, what the hell is going on here? You can make various theories. Like I said, we've tested them all. Maybe those F1 males they prefer to move up the mountain. They're positively phototactic, or they're negatively genotypic. We've tested that. They're not. Maybe they have preference for cold